Hello, Assalamualaikum.
We are
first year
pharmacy student
Of IIUM
are presenting our assignment
on pharmacology.
on anti-platelet drugs.
So let's go.
Assalamualaikum warahmatullahi wabarakatuh. My name is Saiful Bahri bin Suhaimi and I will talk about the history and background of anti-platelet drug. Background of anti-platelet drug. Anti-platelet drugs are intended to prevent and reverse platelet aggregation in arterial thrombosis. It is prominent in myocardial infarction and ischemic stroke. Platelet aggregate will cause hemostatic plaque at the site of vascular injury, and as a result, bleeding is limited and arrested. Massive platelet aggregate will also cause a blockage in human coronary artery. Therefore, platelet aggregation is potentially lethal. Thus, anti-platelet drug was designed to solve this problem. History of first anti-platelet drug. Salicylic acid was the active ingredient from extraction of a plant named willow bark. It is known to have alleviated headache, pain, and fever since antiquity. A French chemist named Charles Gerhardt was the first to prepare acetyl salicylic acid in 1853. The compound he obtained is salicylic acetate anhydride. This preparation of salicylic acetate anhydride was one of the many reactions Gerhardt conducted for his paper on anhydride, but he did not pursue it further. In 1859, von Giem obtained analytically pure acetyl salicylic acid by a reaction of salicylic acid and acetyl chloride. In 1897, chemists work at Bayer AG produced a synthetically altered version of salicin derived from a plant which caused less digestive upset than pure salicylic acid. The new drug from formerly acetyl salicylic acid was named aspirin by Bayer AG. In the 1960s and 1970s, John Vane and others discovered the basic mechanism of aspirin effect. Studies from 1960s to the 1980s established aspirin efficacy as an anti-clotting agent that reduced the risk of clotting disease.
Hello. So moving on, now I'm going to talk about the discovery of antiplatelet drugs. However, I'm going to focus more on the pioneer of antiplatelet drugs, which is aspirin. So enjoy. Aspirin was first discovered in 1897 as an analgesic and antipyretic. After more than 50 years, its ability to reduce the risk of myocardial infarction was found. Later, it was known that the ability of aspirin to reduce cardiovascular disease was due to its effects on platelets aggregation. Here are the chronology of how aspirin was found to be as an antiplatelet drug. First, it was discovered that the aspirin prolonged bleeding time on thrombopathia patients. This may be due to the impairment of platelets aggregation made by aspirin. Second, a research was made by John Vane, Brian Smith, and others, and the result found that the acetyl group in aspirin may play a role in inhibition of platelets aggregation. Later, studies also found that aspirin has also antithrombotic effects, which reduce the extent of thromboxane 2 formation by inhibit cyclooxygenase activity. That is why aspirin therapy is used to reduce the risk of future cardiovascular events, especially myocardial infarction. The discovery of aspirin as an antiplatelet drug has led to many other discoveries of different types of antiplatelet drugs.
Hey, my name is Amirul. I'm going to talk about drug design. Drug discovery cycle begin with compound collection active pharmaceutical ingredient. Then, it proceeds with primary assay, high throughput in vitro method. If no problem detected, it will pass for secondary assay for screen and further for clinical candidate. If not, it will reverse back to designing of the drugs and follow the primary assay again until success. This is method of clinical trials. It goes through all the process until it reaches the final stage and manage to be submitted for trials under controlled variables. These are random trials for aspirin. All 4 types of cancer patients were tested with different dosage consist of placebo, 100 mg aspirin, and 300 mg of aspirin, and the outcome were observed, and also with the follow-up not more than 5 years.
Hi, I'm going to continue on drug trials and we're going to focus on aspirin.
Next, for clinical trials of antiplatelet drug, we are going to explain on aspirin specifically. Aspirin is one of the most researched drugs in the world, and it is estimated that 700 to 1,000 clinical trials being conducted each year. In 1897, Bayer's Felix Hoffmann developed and patented a process for synthesizing acetylsalicylic acid, or aspirin, and the first clinical trials of aspirin began. The results for the first trial were published in 1898, showing the beneficial pain-relieving effects of aspirin. The clinical trials were successfully completed, and aspirin was launched in 1899. After that, in 1974, Professor Elwood discovered first evidence of aspirin's effect in preventing heart attacks. After that study being conducted, larger trials involving 20,000 United States doctors showed that aspirin reduced the risk of coronary thrombosis by 44%. Nowadays, there are many completed and ongoing clinical trials being conducted to study aspirin. You can look up for the trials at clinicaltrials.gov.
Hi, welcome back. So right now I'm going to talk to you about the manufacturing process of aspirin. So we all know that aspirin are manufactured in different shapes and sizes, and it depends on its dosage amount. So now let's proceed to the process of manufacturing hard aspirin tablet, which is known by dry granulation or slugging, as follows. First step, weighing and mixing. First, the cornstarch and acetylsalicylic acid along with the lubricant are weighed separately in sterile canisters to make sure they meet the required predetermined specifications for the batch size or dosage amount. After, the cornstarch is put into cold purified water, then heated and mixed until a translucent paste forms. All the ingredients are spilled into a sterile canister, which is then moved to a mixing machine called Glen mixer. Mixing blends the ingredients and removes the air from the mixture. Then, when the mixture is ready, it is then mechanically separated into units called slugs. Next up, dry screening. After the ingredients have been weighed and mixed well, small batches of slugs are forced through a mash screen by a handheld spatula made of stainless steel. If the batches of slugs are large, they are filtered through a machine called Fitzpatrick mill. The remaining lubricant is added to the mixture, which is blended softly in a rotary granulator and sifter, thus avoid the mixture from sticking to the tablet machine during the compression process. Next, the compression process. In the compression process, the mixture is compressed into tablets by a single punch machine for small batches or a rotary machine for large-scale production. When using the single punch machine, it is put into one tablet mold by a feed shoe, while when using rotary tablet machines, the mixture goes through a feed line into a number of tablet molds which are set on a large steel plate. The plate revolves as the mixture goes through the feed line, quickly filling each tablet mold. Before the tablets get packaged, the compressed tablets are gone through a test. They are tested for hardness, friability, which is to see if they can withstand the rigors of packaging and shipping, and tablet disintegration test. And then when testing is complete, the tablets that passed go to an automated bottling assembly line. They're dispensed into clear or color-coded polyethylene or polypropylene plastic bottles or glass bottles. Then the bottles are topped with cotton packaging and sealed with a sheer aluminum top. It is then sealed again with a plastic and rubber child-proof lid. After that, a sheer round plastic band is affixed to the circular edge of the lid to serve as an adhesion seal to stop and detect product tampering. Bottles are labeled with information of the product along with an expiration date. The bottles are then packaged in separate cardboard boxes depending on the manufacturer. If it is in preparation for distribution for distributors, the packages are then boxed in larger cardboard boxes.
Hello, hi there. You know that there's nothing perfect in this world. The same goes to antiplatelet. Huh? Antiplatelet therapy is unfortunately associated with a risk of hemorrhage or bleeding. But do you know how? Let me explain it to you. Antiplatelet therapy is inevitably associated with a risk of hemorrhage. This is because antiplatelet drugs antagonize platelet activation or aggregation, which increases the risk of bleeding. FYI, even with low-dose aspirin, which is 75 to 100 mg, the incidence of major bleeding has been reported to be 1 to 2%. And most spontaneous bleeding in patients treated with antiplatelet drugs occurs in the GIT tract, with an incidence of 2 to 3% with dual antiplatelet therapy. But wait, what is dual antiplatelet therapy? It is a treatment with two types of antiplatelet agents to prevent blood clotting. The first one is aspirin and the second one is one of 3 P2Y inhibitors, either clopidogrel, prasugrel, or ticagrelor. It is reported that the rates of bleeding increase with the prolonged duration of dual antiplatelet therapy. In the study by Patrono et al., the risk of having major bleeds is 1 to 2 events per 1,000 patients treated. The risk of intracranial bleeding is also increased, with a rate of 1 to 2 per 10,000 patients treated. Oh, and since aspirin is one of the most common antiplatelet drugs, you should know what are the factors that may cause aspirin-related bleeding. First, taking more aspirin than 75 to 100 mg per day. Second, increased age. Third, if a patient has a history of peptic ulcer disease. And lastly, concomitant treatment with NSAIDs, clopidogrel, and also warfarin. So peeps out there, watch out! Okay, that's all. I want to play with my cats. Bye!
Hello guys. Now is actually 2:30 am Baram's DY week. Since I couldn't sleep, I would like to share with you guys about something interesting and knowledgeable, which is antiplatelet drug resistant. In today's video, I will talk about what is actually antiplatelet drug resistant, what is the cause and the setbacks of having these conditions.
Next is the problems with antiplatelet drugs, which is drug resistant. What is antiplatelet drug resistance? From clinical points of view, it is a thrombotic event that occurs in some individuals despite taking antiplatelet medication. For laboratory, it is an incomplete inhibition of aggregation by the medications in question. The factors that contribute to the resistance of the drugs are reduced bioavailability, genetic variations, enhanced platelet turnover, alternate pathway of platelet activations, and individual variation. The reduced bioavailability may be due to inadequate dose and poor absorption of the drug. Mutations in cyclooxygenase 1 genes can be the contributor to genetic variation, which as a result cause drug resistance. Thromboxane synthesis can be induced by other pathway, causing the antiplatelet drugs to be useless, as the drug will usually block the cyclooxygenase 1 pathway only. Diseases such as diabetes, hypertension, and hypercholesterolemia can increase the concentrations of platelets in the blood, causing the drugs to be less effective. The setback of having antiplatelet drug resistance is an increase in the risk of adverse cardiovascular outcomes in patients with coronary artery disease, cerebrovascular accidents, peripheral vascular disease. A recurrent arterial thrombotic event may also happen if the patient is resistant to the antiplatelet drugs.
We'll proceed with the management of gastrointestinal bleeding, also known as GIB, in patients receiving antiplatelet therapy. Firstly, is through proton pump inhibitors. They are taken along with the therapy to reduce the risk of upper GIB, in both low-dose aspirin or LDA and clopidogrel uses. This drug prevents the rebleeding of upper GIB, hence preventing the premature discontinuation of dual antiplatelet therapy. This is because usually after experiencing GIB, many patients stop receiving LDA therapy, which only increase the risk of ischemic events. My next point is endoscopic therapy, which is used to treat active spurting or oozing bleeding or a non-bleeding visible vessel as in the picture. This therapy include the injection techniques using epinephrine or ethanol for hemostasis.
Hello everyone, this is Troya and I'm going to talk about antiplatelet resistance drug, which are aspirin and clopidogrel. So let's go!
Platelet function is regulated by three categories of substances. The first group consists of agents generated outside the platelets that interact with platelet membrane receptors. The second category contains agents generated within the platelet that interact within membrane receptors, while the third group comprises agents generated within the platelet that acts within the platelet. From this list of agents, several targets for platelet inhibitory drugs have been identified. First, the inhibition of prostaglandin synthesis, which is aspirin, inhibition of ADP-induced platelet aggregation, such as clopidogrel, prasugrel, ticlopidine, and finally, the blockade of glycoprotein. First, aspirin. The prostaglandin thromboxane A2 is an arachidonate product that causes platelets to change shape, release their granules, and aggregate. It inhibits the synthesis of thromboxane A2 by irreversible acetylation of the enzyme cyclooxygenase. The FDA has approved the use of 325 mg per day aspirin for primary prophylaxis of myocardial infarction. Second, clopidogrel is frequently preferred over ticlopidine because it has fewer adverse effects than ticlopidine and is rarely associated with neutropenia. The maintenance dosage is 75 mg per day, which achieves the maximum platelet inhibition. The duration of antiplatelet effect is 7 to 10 days. Clopidogrel is a prodrug that requires activation via the cytochrome P450 enzyme isoform CYP2C19. Aspirin and clopidogrel resistance. The reported incidence of resistance to these drugs varies greatly from less than 5% to 75%. In part, these tremendous variation in incidence reflect the definition of resistance, methods by which drug response is measured, and patient compliance. Several methods for testing aspirin and clopidogrel resistance in vitro are now FDA approved. However, the incidence of drug resistance varies considerably by testing methods. These tests may be useful in selected patients to assess compliance or identify patients at increased risk of recurrent thrombotic events. However, their utility in routine clinical decision-making outside of clinical trials remains controversial. A recent randomized prospective trials found no benefit over standard therapy when information obtained from monitoring antiplatelet drug effect was used to alter therapy.